Hi, everyone. I'm Pushpa Itagi, a postdoc from Fred Hutch Cancer Center, Seattle. And today I'll be talking on using multi-omics and liquid biopsies to study tumor evolution and heterogeneity in bladder cancer. Firstly, I would like to thank all the patients and their families for their generous contribution, without which this project would be impossible. I also wish to thank Samantha Schuster, Sonali Arora, and Ming Lam, who are some of the key people for this project. All the members of the Ha Lab and the Shea Lab, the bladder cancer group, and my mentors, Gavin Ha and Andrew Shea. With that, I would like to begin introducing on bladder cancer. It's a highly heterogeneous disease. There is a global incidence of more than 500,000 cases each year. Most of the bladder cancer forms this urothelial carcinoma, or UC, which is the conventional form, but there is other 25% which are more aggressive, and they are of different types, such as squamous, plasmacytoid, neuroendocrine, and others. These are the highly aggressive ones and have poor prognosis. Nearly all the times, bladder cancer in metastatic stage leads to death because there is limited response to therapy. There have been prior studies by the groups such as TCGA, Hartwig Medical Foundation, and others, but they mostly focus on the UC subtype and on the primaries. So we don't have a lot of details on heterogeneity or on the variant level insights. So, a critical need for the field is to understand how these variants evolve and the heterogeneity between patients to help tackle the disease better. For this critical need, we have been working on utilizing this project of rapid autopsy, which began in University of Washington and Fred Hutch since 2015, and it is still ongoing. For this piece of work, we have about 20 patients, and here there are the different histologies, which is determined by pathology. There is the conventional UC and all the aggressive variants. What they have done is at the autopsy, you collect multiple tumors per patient. The multiple tumors would be the primary bladder, a matched normal to exclude all the germline variants, and one or more of these metastatic sites. We also have cell-free DNA collected at the time of autopsy. We have multiple different data modalities. We have whole genome sequencing for 100 tumors, matched bulk RNA sequencing. We have also a subset of single nuclei RNA-seq and cell-free DNA if there was enough tumor fraction and yield. For today's talk, I'll be going mostly on whole genome sequencing and cell-free DNA. To be precise, I have three questions. First is, because we have multiple tumors, what are the patterns of the metastatic seeding that are most prevalent in them? Secondly, if we want to look into heterogeneity, how does the timing of this differ across the patients and across histologies? And lastly, how does heterogeneity is being reflected in the cell-free DNA and how well it captures variant level histologies. For this, we first developed a clonality-based framework. So what I'm showing here is a tree of a patient. The different circles represent different nodes, and the numbers represent different mutations. The first one is the founder clone, meaning these set of mutations are present in all the samples and are clonal. The shared one are present only a subset of these samples and are subclonal. The private are exclusively to just one tumor sample and can be seen as a form of continued evolution. I'm not going into a lot of details, but we developed a very comprehensive framework for this by utilizing all these scholars, such as mutation calling, estimating tumor fraction, and using PyClone, Lychee, and Machina to finally come up with the seeding analysis. I'll bring the same patient again, which is the neuroendocrine patient. What we see from the seeding analysis as shown here is there are these two clones, the founder and the shared clone, that first migrate from the primary bladder to the metastatic pancreas site. And you can see these trees reflect the corresponding trees from these specific tumors. Then the pancreas further develops these two subclones, which are eventually migrated. So at the end of the life, the patient's tumor evolution looks something like this. It's quite complicated because this pancreas further seeds to all of these metastases, such as liver and lymph nodes. We confirmed these findings by cross-referencing to CT scan with the help of the radiologist. So what these scans show is each patient has a scan 6 months and 3 months before their death. So you can see in the first scan, which is 6 months before the death, that pancreas has already been detected here, which is what we observe. That's the first metastatic site. And then eventually all these liver and lymph node mets arise. We did the same analysis for all the patients, and some of the results were quite interesting. The first one we saw is the first pattern was primary to metastatic disease, where these clones migrate from the primary to the metastatic samples. We saw this only in 3 patients. Next, we saw multi-tumor seeding, where the primary seeds mets, but also mets in turn seed each other. But the most common was metastasis to metastasis seeding. So the primary seeds the first met, and all these further mets seed each other. This is the most common one, and also we did not see any correlation specific to any of the variants. But what this highlights is if we target this first metastatic site, then you can alter the course of the disease. With that, we also did another analysis looking at the clonality complexity. What I mean by this is we calculate the average number of the clones that are being migrated. In simple words, the different number of colors in each of these arrows, and then just divide them by the number of metastatic tumors. That kinds of give you some number to address complexity. So we did this for all the patients, took the median, and divided them into two groups, high and low. The groups with the low seeding actually had better survival, and the groups with the high polyclonality index had poorer survival or shorter time to death from metastasis. This highlights that if you have more complex or polyclonal type of seeding, then you'll have shorter survival time once from the metastasis to death. With that, I would like to bring up the next question is how does heterogeneity and timing of acquired genomic alterations differ between patients and histologies? So for this, we again refer to the same categorization that we use, the founder shared and the private clones. We've kept it consistent throughout our questions. And what I'm showing here is a typical landscape plot. On the columns are these different patient numbers colored by the histologies, and these colors reflect the different tumors in each of these patients. We also have a sample level histology, and I'm showing here just a subset of the driver genes in bladder cancer. The top triangles represent the mutations, the bottom triangles represent the copy number alterations, and the open rectangles represent structural variants. We're only showing the pathogenic alterations, which we came up with a consensus calling and multiple approaches. What we see immediately is for P53 and core-R1, which are tumor suppressors, we see it being founder, so they occur much early on. But also, we notice for some specific pathogenic mutations, such as PIK3CA and FGFR3, both of these are clinically actionable in bladder cancer, and you can see these private mutations coming here. These would have been missed if you did single region tumor sampling. So this is a very exhaustive resource to study that there will be later ongoing private mutations, which can actually be more sensitive to therapy. But it also highlights the message that every patient has some sort of pathogenic alterations. And if we just have the primary tumor biopsy, we can't fully capture this picture. Next, we also looked into histology, where we are grouping these by histologies, and each dot represents a patient of that subtype. We again categorized it by founder, shared, and private. And what we see for UC and UCSTs, which again are the less aggressive subtypes, that there's a continued evolution. There's in founder, there's some in private, and there's some in shared. But for PUC and NE, what we saw is most of the pathogenic alterations are primarily as founders. So this disease begins much early on, and later on, it's a much more stunted evolution. We also did some correlation comparing these different types of alterations in the founder and in the private, along with from time from diagnosis to death. And you can see that as expected that if you have more founders, you have lesser time to death. And then we also saw one more interesting observation that is you accumulate more private mutations as the patient lives longer. But eventually, both of these subtypes of aggressive and the less aggressive get killed, but just by different kinds of alterations. With that, I would like to move to my last question, which is understanding how does cell-free DNA capture genomic heterogeneity and transcription factor profiling. So cell-free DNA offers a minimally invasive window into tumor biology, and our analysis mostly focused on circulating tumor DNA, which is the tumor-derived component of the cell-free DNA, enabling a sensitive and minimally invasive way to track clonal heterogeneity. So again, we have patients collected blood or plasma or serum at the time of autopsy, and this is just a schematic representing how their tumor evolution trees were. We were expecting to see some contribution, hopefully more of the founder and less of the shared and private, and there will be some contribution from the immune cells or the non-tumor cells. We had 8 samples which went through whole genome sequencing because they had high enough tumor fraction, and we had 17 that we went for targeted panel of about 25,000 X coverage for this 400 genes. So looking at the total capture, what these box plots represent is 3 different groups. Each dot is a patient. And we look at the proportion of tumor-matched single nucleotide variants that are captured. So of all the tumors, how many of those or what proportion of those can the cell-free DNA capture? It looks close to about 75% for the whole genome sequencing and targeted panel. It does a little poor for the low tumor fraction ones, but it still does some capture for it. Once we start looking into clonality and we go into the founder shared and private status, we see some nice results, such as we have nearly 100% median capture for all the founder events. So if you have a pathogenic event that is a founder, your cell-free DNA nearly captures most of the times. And then there is a slight drop for the shared, and then a few further drop for the private. We did further analysis on this, which I'm not highlighting here, but mostly the private mutations lower capture is because it's dependent on the tumor volume, the shedding rate, and some other factors. But we also see that even if we have lower tumor fraction, less than 8%, we can still get some capture with it. And then nextly, we also looked at transcription factor activity in the cell-free DNA samples. So what I'm showing here is a schematic from a tool called Griffin, which was developed in the Ha lab. What it does is it looks into loss of coverage at these certain sites. So if we have a nucleosome depleted region where a transcription factor binds, then there is loss of coverage in that region. Whereas if there is no transcription factor binding, then it is nucleosome rich, and then we won't have this loss of coverage, but you rather see this flat profile. So what we aim to look is to distinguish between healthy donors and the metastatic bladder cancer, or between the bladder cancer subtypes. We're looking for this dip or this trough or this loss of coverage. So as the first proof of concept, what we did was we took healthy donors, about like 6 patients, and 8 of these whole genome sequencing bladder cancer patients. And we applied this tool and utilized the attack-seq sites specific for bladder cancer from this paper. And what we are looking is if we have higher nucleosome coverage or less accessibility for the transcription factor, we see this flatter profile. And if you have lower nucleosome coverage or a nucleosome depleted region, then it is more accessible. So this clearly shows that as a proof of concept, you can determine from the plasma or the serum that this is a metastatic bladder cancer sample. We also did some subtyping. So we looked into this classical neuroendocrine marker called as ACL1. And you can see that in this one patient, which is neuroendocrine as determined by pathology, that it is more active and has lesser coverage because the transcription factors specific to this subtype are active, whereas all the other subtypes, such as UC and PUC and healthy donors, are much more here and much more flatter because they're not active in this subtype. With that, I would like to conclude and reiterate on the summaries. Firstly, on the metastatic seeding, we see that metastasis to metastasis is the most common. And also, if we have early metastatic intervention for the first metastasis, then it will likely alter the disease course. And if we have higher complexity, then we'll have lesser survival time after developing the metastasis. Looking into the tumor evolution and genomic heterogeneity, we observed that every patient has some pathogenic alterations. Mostly the aggressive types, such as PUC and NE, have early evolution, and UC and UCSTs have more continued evolution. This also suggests that all of these subtypes should be focused differently for therapy. We can't use the same type of therapy for all the subtypes of bladder cancer. And lastly, from the cell-free DNA, we were able to capture 100% of the founder mutations, and we could use sites specific to bladder cancer to distinguish it from the healthy samples, and also do subtype-specific subtyping, looking at the transcription factor activity. With that, I'd like to thank all the members from the Ha and the Shea Lab and the ISMB organizers for giving me an opportunity to present. And thank you for your attention.
Thank you so much for such impressive work. Any questions?
Hi, thank you for a super clear talk. It was really fantastic. I had a couple questions. So with the metastatic seeding patterns that you looked at with Machina, were you able to see a difference in the prevalence of different metastatic sites in the different types of patterns? And did this vary amongst the different cancer subtypes?
That's a good question. We did check into that, but we didn't see anything different. Generally, we have enrichment of more liver subtypes and lymph. So we also had some sort of low N, but it was not specific to any particular subtype.
Okay, very
Sorry,
interesting.
the
And
tumor
then
location.
the second quick question is for your cell-free DNA analysis. Did you, I didn't quite catch it, did you see a relationship between the variants you were looking at and their clone proportions in the samples that you had?
We didn't look variant specific for the clone proportions, but we looked overall for all the samples. And we discovered that Y shared or private could be lower, whereas the founder is higher. But again, it was not specific to a subtype. And we just had like 18 samples in total, which becomes difficult to
Right,
assess.
yeah. Thank you so much.
Hi, great talk. Just a couple of questions to follow up on that. In the first part of your talk, since you don't know actually when the metastases pop up, did you find any pattern that break the logic of the tree that you built through the mutation? So because those patients sometimes they go through some specific treatment, right? And those treatment there is selection pressure which the tumor is trying to escape. So you might have two different metastases that appear, but then the same gene variant of the same gene is affected. So they're trying to then being activated, although they're not one child of the other. You know what I mean?
Yeah. So the tool Machina does give you multiple solutions. And there were cases where two tumors were likely to seed or be the primary seeder, which becomes confusing because they have the same set of alterations. For that, we cross-referenced to CT scan to see which one of those came first in the scan and took that as a primary seeder. But yeah, that can happen.
Okay. Yeah. Then following on the same similar question, the cell-free DNA portion, you see the primary bits more present because it's just there are cell or the VAF, the frequency of this mutation, you see more because they are shared by everyone, right?
Right.
And of course, we also might see some specific location shed more, maybe because it's in the liver, which where this end up the blood being going to, right?
Right.
Other one are lost.
Right. We did see, it's more in our supplementary work, that liver sheds a lot, especially if a patient has like 2 to 4 metastases, then those patients actually shed a lot of cell-free DNA, and particularly ctDNA.
Hi, so I have a more general question. So given all this great work you've done, how did it change how do you think about the therapeutic strategy given all this work you've done? How does it change the...
Right. That's a great question. I think it should lead more to like personalized medicine depending on the subtype.
Strategy focus on
Right.
on this metastasis? Is other mechanisms that are currently targeted should be are better candidates, or?
Right. So there are we did a lot more work, and there are certain USP1 inhibitors which are not used in bladder cancer, which is in one part of our work, which could be used because we see one of these subtypes having elevated DNA repair pathway. Or also, if for the figures that I showed, we could see that PUCs and NEs, you might want to target just the founders, whereas for UC and UCST, rather than being more targeted, you need to go a different way. So a lot of these are actually currently using targeted therapy or like ADCs, but they have to be more specific. And I think we need more data to go directly into that kind of route. But there have been other studies in the department that are going on that, like MVAC trials and others.
Any other question?
Thanks for the talk. Really great work. So in your presentation, you had these two models of evolution. One subtype or with the continued evolution, and the other with the stunted. So did you conclude that was because the ones with the stunted evolution, they are so aggressive that they don't get the chance to perform the continued evolution? Was that the conclusion from your analysis?
So we went the other way around. We know that they're aggressive and they die sooner, but we didn't know what was the cause. So when we see a lot of founder mutations or alterations, which are present in all the cells, so likely those arise much earlier than the less aggressive. So that is what kind of kills that subtype. So you'll have to target the founder alterations there rather than very private or subclonal ones.
Any other question? Well, in that case, I just leverage for a quick question too. I wonder, in your cohort, the primary tumor is in situ during autopsy, right? So did you see
Right.
any evidence of like, receding of the primary tumor?
That's a good question. So a lot of these primaries are taken before autopsy during like TURBT or cystectomy, and some of them are taken at autopsy. So we just had like 7 patients with taken at autopsy. We didn't see any reseeding in them, but they do have like private mutations or alterations to them, like KMT2D, which is like a result of continued evolution. Also, I think one of the limitations is we don't have multi-regional sampling of the primaries, so maybe that would help to highlight if there is more reseeding.
Wonderful. In that case, we can thank the speaker again for such a fantastic talk.